This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 840, recorded on December 9th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Omicron.
Yes, we will talk about Omicron, however you want to pronounce it. But let's get right into it. Let's start with the quotation, because we actually have a lot to cover today. Oh my gosh, we're at Clinical Update 92, TWiV 840, and still there keeps being a fire hose of information every week. So, a smooth sea never made a skilled sailor. And that's by FDR. And there has been no smooth seas over the last 2 years. This has been quite a challenge, but I am hoping, I'm optimistic that we're getting better. But let's talk a little bit about Omicron. So far here in the U.S., we're still seeing trouble with Delta. We saw that post-Thanksgiving surge in infections, and then that was followed by a surge in deaths. We actually had a day here in New York where we had over 100 deaths in a single day. We are starting to come off that hump here. We'll see what happens, because we have another holiday coming right up. So we'll see where we are there. But let's start talking a little bit about Omicron. So first, I'm going to start with a preprint. And don't worry, you're going to get bad news. You're going to get good news. I think it's going to work out in a positive direction in the end. But this first preprint: increased risk of SARS-CoV-2 reinfection associated with the emergence of the Omicron variant in South Africa. Now, this got a lot of attention, and here the authors are reporting on a retrospective analysis of routine epidemiological surveillance data collected through South Africa's National Notifiable Medical Condition Surveillance System. So basically, what they're doing is they're looking at individuals that had a positive test, and then they had a second positive test. It had to be at least 90 days apart, right? So this isn't just someone who has a string of positive tests. This is suspected reinfections based upon that criteria. So they looked at 35,670 suspected reinfections. These were identified among 2,796,982 individuals with laboratory confirmed SARS-CoV-2, right? So just, I remember, Vincent, it was not that long ago, there was this whole debate about, can you get reinfected? Mm-hmm. And in this one study, we have 35,670 reinfections meeting this criteria. So if there was any doubt, you can get reinfected. And what they reported was that the recent spread of the Omicron variant was associated with a decreased hazard of primary infection and an increase in reinfection hazard, right? So of the infections they're seeing here, they were seeing about a 2.39 increase in the hazard ratio for reinfection. So just sort of as far as what did they conclude, what do you take away from this? Quote unquote, I'm going to read, population level evidence suggests that the Omicron variant is associated with substantial ability to evade immunity from prior infection. In contrast, there was no population-wide epidemiological evidence of immune escape associated with the Beta and Delta variants. I think this is interesting and concerning, but let's not just sort of do what we do, with vaccines, right? We make a distinction. What we want to know is not only can these individuals get reinfected, but we're also going to be interested in how do they do. Are they just getting reinfected? Is breaking through that initial line, but are they still protected against moderate to severe disease? Mm-hmm. So I think that's going to be, coming with, data that comes in the future. A couple of the things I thought were interesting is in this data set, there were actually 332 individuals who had 3 infections. It was even an individual who was infected for the fourth time during November. So not only do we see reinfections, but we see reinfections and reinfections. So
But
I'll put
Daniel,
this.
if you
Yes,
did this
Vincent.
kind of a careful study elsewhere earlier, maybe you would find the same thing, but we just didn't do it, right?
Yeah, no, I think that's important, and I think it's unfortunate that we don't have the data at this point because we don't just want to say like, okay, you've been infected, you're at such and such risk of reinfection. We also want to know how do you do, right?
Hmm.
Because at some point,
Sure.
I mean, this is a virus that is here to stay, and so we need to know these things.
Well, Daniel, the common cold corona, the study of Jeff Shaman showed that people get multiple infections even within a year. They don't even know it because they're so mild or asymptomatic. So this is not necessarily a bad thing, right?
Yeah, and I think that's really important. I still remember, and I emailed Jeff early on in the pandemic talking about this, and he's been on actually. I think he's been on TWiV, and
Hmm.
that's important to know. Let's think about a future when we are either vaccinating or not vaccinating, and a person either gets infected when they're really young or gets vaccinated and then gets infected. Maybe they keep getting infected every year. If it is nothing more than the sniffles, that is a much different world that we're
Hmm.
living in than we're living in right now. All right, children. Children are at risk from COVID. I think this is right up there with reinfection. We now know this happens. Children get infected. Children actually, unfortunately, some of them can end up in hospital, and some of them can end up dying from COVID. There was an expansion of the EUA for the Eli Lilly monoclonal cocktail all the way down to newborn. So let me just go through this. The US FDA revised the EUA for the Eli Lilly cocktail, bamlanivimab and atezolizumab, to additionally authorize the administration for the treatment of mild to moderate COVID-19 in all younger pediatric patients, including newborns who have a positive COVID-19 test and are at high risk for progression to severe COVID-19, including hospitalization and death. So think about, we've talked about this before, congenital heart disease, immunodeficiencies, the requirement to be on immunosuppressive medication. It used to be that there was a certain age and a certain weight cutoff, but now, at least for Eli Lilly, we can actually extend access to this therapy all the way down to newborn. So I think that's a bit exciting, and there is even more exciting monoclonal updates to come. Pre-exposure transmission testing. I like to say never miss an opportunity to test. I'm going to go a little off script here. There was a White House press conference, right? I don't know if you, I saw that. I wasn't watching it live, but I saw afterwards, and someone was asking about testing. And the Jen Psaki sort of responded, what, are we supposed to mail out tests to everyone? Well, the quick response on the internet was yes. Yeah, and I do think there is a hunger in the United States. There's an appreciation that testing is a really important way to keep people safe, especially with the upcoming holidays. Vaccination reduces your chance of becoming infected, but you still can get infected. You still can transmit. Testing really adds another layer. So yes, we do need rapid tests out there. Let's flood the U.S. with rapid tests as opposed to what we're currently seeing is the U.S. being flooded with COVID. All right, back to serology tests. And I want to reference the recent TWiV. Actually, I just enjoyed that this afternoon. I carved out a little time as I was driving from hospital to hospital. I listened to it on 2X, Vincent. So
Mm.
when you and I talk, it's funny because my kids are always, why does Vincent talk so slowly? Because even they're used to hearing you at 2X.
Oh my gosh.
But
Can you understand it at 2x?
I sort of, there's a moment I have to shift over, but
Huh.
I hope I understand
Wow.
it. But what I thought was really important on that episode, and this is something that we really are going to want, and as I go forward, you're going to understand why we're going to want more of this, is, right now, everyone and their uncle makes a serology test for COVID, right? So I get patients, oh, I got this test and it was negative. I got this test and it was greater than 250 and this other test. We should be able to reach a point where we have serology tests, neutralization assays that we can use as clinicians to get at least some sense something we can use clinically to decide at what risk an individual is. We have all these serology tests where a person was infected and then they got one vaccine, maybe they got a second vaccine. They're trying to decide, am I supposed to get boosted? Do I need to get a third vaccine? We have individuals that got one shot, a second shot. They're trying to make decisions about that third shot. Maybe they had difficulty with that second shot. At some point, maybe people are going to be talking about fourth shots. It would be nice for certain individuals to actually have access to a reliable test that does correlate. So sort of this is my everyone listening. We need those tests. I think as clinicians, a lot of clinicians are out there using what's currently available. It's all over the place. We don't really have a tight correlate with immunity. There certainly are research assays and studies suggesting that this is something that can be done. So yeah, we could use those tests. That would really help us. I think it would also help address this question. We don't necessarily want to know on a population level, but if someone was infected and they had one or two shots, it would be great to be able to help guide them. Do they need a third shot? Do they even need a second shot after that first one? So Vincent, I don't know if you had any thoughts. I got to hear all your thoughts, and I'll recommend everyone listen to that TWiV. But
No, I agree. I think we need a standardized test and that can be used by everyone. And I don't know that will happen quickly, right, Daniel? It takes time.
yeah, it will take time, but I think it's a priority. I think there's a lot of individuals out there, unfortunately, who've been infected, who are trying to decide about vaccination, how many doses, what do they need to do. I think Omicron is a big push, right, to go ahead and get at least one or two of those vaccine doses. Do they need a third? We'll have to see, but maybe they don't, and I think that'll be important to see. All right, active vaccination, right? I'm talking about vaccines, and now we're getting right into it. Never miss an opportunity to vaccinate. We're all starting to hear about the effectiveness of our vaccines against Omicron. There is some data starting to become available. And so the investigation, SARS-CoV-2 Omicron, has extensive but incomplete escape of Pfizer BNT 162b2 elicited neutralization and requires ACE2 for infection. This was sent my way. I think, Vincent, you may have even shared it with me.
I think it was Amy.
Was it Amy who shared it? Okay, excellent. And thank you for all the people that keep sharing, preprints or even stuff that isn't even preprint. It's great to try to be able to keep up on this, and it helps when all my colleagues. So, Amy, thank you. So here the investigators used early passage of isolated and sequence confirmed Omicron variant virus isolated in South Africa. So they're using the virus. This is not a pseudotyped assay. They used a human lung cell line clone engineered to express the ACE2 receptor to both isolate the virus and test neutralization. And so they were able to demonstrate that the Omicron variant virus was still using ACE2 for entry. They tested the ability of the plasma from the BNT 162b2. So these are the Pfizer vaccinated study participants to neutralize Omicron versus the ancestral D614G virus in a virus neutralization assay. Beautiful figures, right? So if people read this or see figure 1 shared on social media, I've seen it shared on social media, they are using something called an FRNT50. So this is a focus reduction neutralization test. It's somewhat similar to plaque assays. It's been suggested that it is easier. It is less labor intensive. I see Vincent shaking his head.
Easy way out. Always taking the easy way out, folks.
Okay. Well, they went ahead and they tested plasma samples from 12 participants that had all been vaccinated with the Pfizer vaccine. Now, 6 of them had no prior evidence of SARS-CoV-2 infection, right? So they just getting the two shots. The remaining 6 actually had a record of previous infection, right? So they were infected with the ancestral D614G, and then they got 2 shots on top of that. So what did they find? So they found that there was a 41-fold decline compared to the only 3-fold decline seen with the beta variant as far as this neutralization. with the initial neutralization being so elevated with the 3 exposure scenario versus the 2 vaccine, when you actually look at the drop, you drop to a level that was actually higher than we were seeing with the 2 doses of vaccine. So the escape was incomplete. And so even after this drop, there's sort of the suggestion that 3 exposures might get us there. And I'm gonna get into that next. So what about 3 exposures of vaccine? What if instead of infection and 2 doses of vaccine, you get 2 doses of vaccine and then another dose of vaccine? So I haven't seen the raw data on this, but here we're talking about a 41-fold drop. We are seeing, or we are hearing from Pfizer, and again, I haven't seen the raw data on this, a 25-fold rise with that third shot. So really getting us back up to a level where we anticipate that 3 doses of the mRNA vaccine are actually going to continue to give us protection. So the folks at Pfizer are suggesting that since this is the case, that we should feel a little bit better. But Vincent, what are your thoughts on getting a booster after hearing this?
So we did a paper on TWiV, which you listened to, where a group in Canada showed that if you spaced dose 1 and 2 16 weeks apart, you get a much broader antibody response than if you space them 4 weeks apart. And so it seems to me that a third dose would correct for that inappropriate early spacing. And so it makes sense to me now to get a booster to broaden those antibodies. And I think it would include Omicron as well. I don't see why it wouldn't. So now I am slowly seeing the data that a booster is probably a good idea for that reason.
Yeah, I mean, I think this is really interesting. I remember John Muscola and I were emailing a bit, like when the boosters were first being rolled out, I guess, to the general population. And there was this idea that really the mRNA vaccines were turning into a 3-dose primary series. And then, so maybe that third dose, really, boost isn't really the right word. It's really just you're finishing off
That's
your
right.
vaccine. You're getting that breadth. You're getting that high level. It is interesting, this discussion. Like, if we could go back and do all the science again, would we test 1 dose, wait 3 months, 1 dose, wait 6 months? And the logic was, it still makes sense to me, is you give the first dose, you give that second dose 3 to 4 weeks later, you get up to a high level, you've got a degree of protection while you're waiting for that third dose and that broadening and expansion. So it makes a lot of sense in the midst of a pandemic with high transmission. And I don't know if we'll go back. I think it'll be much like hepatitis B, where, the standard is 1, dose at time 0, another dose at 1 month, another dose at 6 months. But that being said, there is now a 2-dose hepatitis B vaccine out there. So, we'll see what time.
But Daniel, it could be that 2 doses 16 weeks apart is as good as 3 doses, right? When the first
Yeah.
2 were too close. So I wish they'd do a larger trial, especially in 5 to 12-year-olds who are just starting, right? Getting their 4-week spaced, and maybe they shouldn't be.
It is interesting. And, and I think if we circle back to having a really reliable correlate of immunity, a serology
Yeah.
test, a T cell, you don't
Yeah.
need huge numbers to redo that. And it does help. I mean, we're still in a pandemic. It would be nice to know. Also, it would be nice to know as we come out of this, if this becomes a routine childhood vaccine, do we just do it 2 shots quote unquote properly spaced? I mean, that science would be helpful. All right, passive vaccination. This is exciting. This is big. We now have monoclonals for pre-exposure prophylaxis. So this hot off the press, Wednesday, December 8th, the FDA announced EUA for AstraZeneca's, and they named it Evusheld. I think it should have been Evushield or something. I should. You can call me. I'll help with the names. But this is a combination long-acting antibody cocktail, tixagevimab, co-packaged with silgivimab. Okay, rolls off the tongue. And this is for pre-exposure prophylaxis for COVID-19 in certain adults and pediatric individuals. So this is going to stop there right at age 12 and a weight of 40 kilograms or 88 pounds. Now, the product is only authorized for individuals who are not infected, not currently infected with SARS-CoV-2, um, not right after a recent exposure. This is upfront. You plan this ahead of time. This is for individuals who either have moderately to severely compromised immune systems due to a medical condition or due to taking immunosuppressive medications, or they have a history of a severe adverse reaction to a COVID-19 vaccine, right? So this could, in certain individuals, let's say they get that first dose and they develop a vasculitis or some other really significant adverse, we're not just talking about a rash or something that we could get through. This can be used in both those contexts. So there's a list of who are these individuals that are moderately or severely immunocompromised. So active chemo treatment, right? So active treatment for a solid tumor or a hematological, that'd be a blood cancer, a person who's actually received a solid organ transplant. I think there was a discussion of the West Nile transmission in transplant patients, an individual who's on immunosuppressive therapy, and the list sort of goes on to include a lot of other immunosuppressive conditions. So there is a huge need for this. And I think we've talked about this before. There are individuals who cannot get protection from the vaccine or not get the level of protection we would like to see. And those individuals have been basically living frightened, a lot of them afraid to re-enter society because they have been vaccinated, but they don't have B cell function. They have other issues. So this is huge. This is really big. But still remember, this is a pre-exposure prophylaxis. It's going to be a twice a year injection. We also still have our post-exposure, and we have our early treatment with the other antibodies out there.
Daniel, are these two monoclonals able to neutralize Omicron?
I would love, and maybe our listeners can help us with this. The company has not released data yet. They're working on it. They're testing this. One of the things I do not know is I don't know exactly where on the spike protein these two antibodies bind. I have the maps for all the other antibodies, and I could not find this information. So, if anyone has it, it would be nice to know, like up front, where it's targeting. Is it outside of this rather variable receptor binding motif, like the Vir GSK sotrovimab, which looks to still be effective? So I think that's going to be really important if Omicron moves into our region. How much protection will these individuals have against that variant? So excellent question. And yes, if any of our listeners know a little bit more, I would love to know. All right, the period of detectable viral replication, right? So now an individual has tested positive, and I'm still saying this is the time for monitoring and monoclonals. We're still waiting on those oral therapies that might enter into this region. But now we've entered what I call the battle of the monoclonals, which is the best monoclonal. So we had the real world assessment of 2,879 COVID-19 patients treated with monoclonal antibody therapy, a propensity score matched cohort study. This was published in Open Forum Infectious Disease. And the authors reported on 4,328 SARS-CoV-2 positive patients who were treated with monoclonal therapy. And I'm just going to cut right to the chase. Among the infused cohort, all of them did significantly better than the control, the matched controls. But those who received the Regeneron cocktail had the best outcomes. They had a significantly decreased rate of admission relative to the other groups. And that was an adjusted risk ratio of 0.51. So pretty impressive. And another preprint, and I think this is going to be exciting too. A lot of exciting stuff here in the monoclonal area. The preprint association of subcutaneous or intravenous route of administration of casirivimab and imdevimab monoclonal antibodies with clinical outcomes in COVID-19, right? So one of the things that has stood in our way, an operational challenge with monoclonals, is having to put in and maintain an intravenous line. So this was the question: can we just give them subcutaneous? Is that going to be as effective? So here we get the results of a prospective cohort study of outpatients with mild to moderate COVID-19 symptoms who were eligible for and received monoclonal antibody treatment under the EUA. And they either received the subcutaneous or the intravenous administration of the Regeneron cocktail. So that's the casirivimab and the imdevimab. And they found that both routes of administration were very effective with no compelling differences in effectiveness. So I think that's exciting. There's sort of in people's mind that, oh, if it's intravenous, it's going to be that much better. It looks like subcutaneous can be very effective. There doesn't tend to be a big downside. And the long-acting antibodies, those might be given intramuscular. So we're starting to see more flexibility, more operational flexibility with getting monoclonal subcutaneous, intramuscular. A little bit of an update for in this zone. So people may share this. One of the things I've been doing through the IDSA is helping out with clinical care in Bangladesh, in the Rohingya refugee camps. And so it's pretty exciting. I get on a call, there's maybe 20 or 30 clinicians. We use Teams, sort of technologically allowing us to connect. And we discuss challenging cases in their management. And one of the things that came up recently was, what about convalescent plasma? Like, should we try to mobilize and get that in these refugee camps for the early treatment? And just an update, the WHO COVID treatment guidelines were recently updated, and they now have a strong recommendation against using convalescent plasma outside of a clinical trial. And they go on to say, evidence has not shown the costly, time-consuming transfusions to be effective. The guidance against the use of convalescent plasma was published in the British Medical Journal and was based on the review of the results from 16 trials involving more than 16,000 patients. So there was a lot of excitement here. There were all these ideas that you would get, artisanal convalescent plasma right from people in the local area with the right variant, and really some passionate people. But, we've spent billions of dollars looking at this. It just does not look like it's making that difference that we hoped it would. So really think of monoclonal antibodies as the evolution of this idea into something that really does make a difference. All right, the early inflammatory phase, right? So this is often when an individual might get hospitalized. We've sort of got this outline, hopefully, in people's head where during that first week, the viral symptom phase, this is when you start seeing that early inflammatory phase. This is when you start to see in some individuals that oxygen level drop below 94%. Now, this is typically the second week, but remember, this is a crude model. Everyone's a little bit different. So particularly in older individuals with comorbidities, we might see that drop right around day 7. It might be right the last day or two of that first week. Lower risk, younger individuals, when we see them decompensate, dropping their oxygen, it might be closer to day 13 or 14. So you want to sort of be putting it all together. When was symptom onset? That's the history. I like to say the most important but least reliable. What is that oxygen level? Sort of putting the pieces together. So this is when we start thinking about adding dexamethasone, that 6 milligrams a day for 10 days in individuals with oxygen saturations less than 94. Sometimes we can keep them out of the hospital with that. Often here in the US, we're using remdesivir. Maybe not as much as we were early on, not as much excitement, and the data is not getting us excited. And this is also when we think about anticoagulation. So today, let's talk about that. There was a recent review, and this is this question of, now that we're doing a lot of things to these patients, including giving them steroids and immune modulators, does that impact that clotting risk? So this was the review article, review in patients hospitalized with COVID-19, the effect of adding immunomodulatory agents to usual care on thromboembolic events was assessed. This was published in the Journal of Clinical Medicine, looking at articles that address this concern. They ultimately analyzed data from 8,499 patients, where 4,638 were treated with an immunomodulatory agent. 3,861 without an immunomodulatory agent. Lots of obvious issues with these two groups. Why was one treated? Why was one not treated? Who was their doctor? So these are not going to be perfectly matched. But what did they find? They found that among the patients prescribed immunomodulatory agents, there was a rate of 1.77 VTEs per 100 patient months compared to 2.30 among those treated with usual care. So there was an odds ratio of 0.84, so it does look like a little bit of a reduction, but wide 95% confidence interval. So this did not reach statistical significance. So you see that we always talk about a trend, but a trend that does not reach statistical significance is something that did not reach statistical significance. And basically, I'm going to just sort of conclude with, as they say, they did not identify a statistically significant effect of immunomodulation on thromboembolic events in COVID-19. So in many ways, this can be helpful because there's been a lot of trials looking at anticoagulation in the setting of COVID, and a lot of concerns that now that the standard of care has changed so much, can we still use those data? Have the baseline risks really shifted in a measurable manner? And this is, I'll say, reassuring that if anything, things look like they might be going in the right direction, but not in such a huge variation that we have to repeat all those trials now. All right. And I am going to go right to the tail phase, long COVID, post COVID. And, as I think has become really clear, even when I talk to patients that are not comfortable and excited about vaccination, people appreciate long COVID. This is something that I think we all agree on at this point. Maybe not us all, but most clinicians and a lot of the public are aware, know people who had COVID who did not get better after just 2 weeks. And so the preprint reduced incidence of long COVID symptoms related to administration of COVID-19 vaccines, both before COVID-19 diagnosis and up to 12 weeks after, was posted as a preprint. So this was a retrospective analysis of the medical history of 240,648 COVID-19 infected persons that were identified with factors influencing the development of progression of long COVID. So this analysis revealed, and I think this is actually pretty exciting if this pans out at the end of peer review, that patients who received at least one dose of any of the 3 COVID vaccines prior to their diagnosis were 7 to 10 times less likely to report 2 or more long COVID symptoms compared to unvaccinated patients. So this tends to reflect what we're seeing, right? And I brought this up several times that we are not seeing a lot of individuals who were vaccinated and then got COVID end up in our COVID recovery center. So this is, I'll say, a little bit more data to support that observation. Now, furthermore, and this is where I think it gets really exciting, unvaccinated patients who received their COVID-19 vaccination within 4 weeks of that initial infection were 4 to 6 times less likely to report multiple long COVID symptoms. And those who received their first dose 4 to 8 weeks were 3 times less likely than those people that continued to remain unvaccinated. So we're starting to get a robust, right? This is a data set, 240,648 patients analyzed. We are seeing that vaccination before was associated with a decreased risk. Vaccination within 4 weeks really dropped that risk. Getting vaccinated within 2 months still dropped that risk. What we had seen from other studies, where if you really wait months out, you may only drop that twofold. But there's a couple challenging and interesting things in this paper. One is the issue of defining long COVID. So I like the fact that they looked at a constellation. You needed to have 2 or 3 or more long COVID symptoms. Because if you just start looking at single isolated things, are you anxious? Are you depressed? These things have such a large prevalence, high prevalence in the community, that it's hard to tease those out. So I did look over another preprint that was reporting that vaccination does not protect against all diagnoses identified under the PASC umbrella. And just to, I think, put this in context with the paper we discussed, they looked at 45 different diagnostic codes and reported that only a little more than half of them were significantly reduced by being vaccinated prior to infection. I guess I would report it the other way. I'd say, more than half of these individual codes were decreased. There were some challenges in this, but I think it's really important and a challenge when we look at post-acute sequelae of COVID to really understand that it is a challenge to identify this cluster of symptoms that identifies these individuals. And I will say, vaccines are helpful, but as you're seeing here, it was a risk reduction. The vaccines are not 100% at preventing long COVID, and getting vaccinated after you develop long COVID is not 100% at getting rid of that. We still need a lot more research to help these individuals. No one is safe until everyone is safe. Looking at the global picture, I am hoping that at minimum, Omicron is a wake-up call to the priority of vaccinating the world. We're now all talking about how we need to get that third vaccine to protect us against Omicron, a country of 350, almost 400 million here in the US. What if we had just gotten everyone in South Africa vaccinated, right? That's less than 350, 400 million folks. We really need a global plan. It's not as simple as just sending a whole bunch of boxes of vaccines. You need a plan to get those vaccines into arms. I'm going to steal a tweet that I saw by Roxanne Comsey. I don't know if she originated this or someone else. We need to get the nation out of vaccination. We need to make this a global effort. And this is going to be working with everyone. This is going to be working with those large pharmaceutical companies. They're our friends right now. Governments, maybe they're our friends right now. Grassroots organizations, we all like them. And you need people who have relationships, because as we saw, even in South Africa, the vaccines were arriving, but you need education, you need communication, you need trusted people there on the ground talking with people. So let me get to the last and our fundraiser. Let me remind everyone to go to parasiteswithoutborders.com, click on that donate money, that donate button, so you can send money to us. Thank you, actually, for all the people who have helped us continue to do what we do. Through the months of December and January, we will continue to match those donations up to a total of $40,000 to send to Microbe TV. We may even give a little bit more to them because we have such a great relationship. And if you send more than $1,000, send an email to Vincent at Microbe TV. Let us know, and he's going to get a book out to you. And Parasites Without Borders is going to cover those shipping costs. If you're here in the U.S., just your mailing address. But if it's overseas, throw in that phone number as well. Thank you so much for all your continuing support.
Thank you, Daniel. It's time for some email questions for Daniel. You can send yours to daniel@microbe.tv. First from Jack. I was taking an immunosuppressant drug, Enbrel, at the time I received my COVID vaccines. My booster was in August. I stopped taking the drug in November. Does this mean that my immunity protection from COVID should be at the same general level now as a person who was not immunosuppressed?
So this is interesting. So when you get vaccinated relative to when you're on immunosuppressives, if you're on immunosuppressive when you get vaccinated, the concern is that you're not going to develop as robust a response to the vaccine. So then if you, let's say you stop and you've been off it for a while, it isn't as though suddenly that immune response ramps up. So there's two issues. This one is the potential impact on when you were vaccinated. The other is whatever recovery from being off the therapy. So Enbrel is something that probably after about a month, it's really losing its impact on the immune system. So by now you're pretty far out from that. Boy, isn't this great situation where you'd love to be able to go get a test to see where you are, a reliable test that correlated with protection to determine whether or not you needed another shot to get up to that higher level.
Daniel, that test would also be helpful for people who have been infected and recovered to say, hey, I have the approved level of immunity.
I think that would be huge, right? I think that would be huge for breaking down the division, right? There's a lot of people and they're frustrated and they talk about, why are you not recognizing? The only downside to recognizing that level of protection would be those people who might go out and go to a infection party to somehow get out of a mandate. Don't do that. But I do think a larger group of people are responsible and they've been infected and they're really just trying to decide, what is the best thing for their health.
Our next question is from Amanda. I'm fully vaccinated, received Moderna in January and February 2021. With my second shot, I had a mild reaction 20 minutes after administration. Pretty intense itching around my mouth, inside my mouth, tongue, tingling on my tongue, no swelling, rash, or hives. Went away after 2 hours. I also experienced some heart palpitations, tachycardia for a couple of months after this. Having tried to do my own research, I failed at finding articles about reactions only after the second Moderna. They all concentrated on after the first. My primary care physician has advised me not to get a booster, saying my risks outweigh the benefits. I'm curious if your advice would be the same.
You know, the immediate reaction around the vaccine that you described, that's something where usually I would have someone see one of our allergists. Actually, David Wertheim is an allergist here at ProHealth. That he and I, boy, we have a relationship now. We barely communicated before, but now, you know, a lot of times the allergists can help, and they'll do sort of peri-vaccine medications to help control some of those responses. I'm a little bit more interested in the late manifestations, what you talk about with what might be cardiac manifestations. So that's something where I would probably want a little more information. You really want to make sure that you're not going to have an issue there. And again, we're back to, it would be great to get a sense of where you are, because the alternative here is potentially doing something like the AstraZeneca. If there really is valid concerns that that second dose may have caused some cardiac issues, and we're concerned that a third dose would trigger that again, then it wouldn't be do nothing. It would be look at getting you that protection, because if this is somehow related to an immune response off target from the spike, boy, letting you actually get infected with SARS-CoV-2, that may be a bit of a disaster.
Marshall from Austin writes, my son, 10 years old, no pre-existing conditions of concern. He's back in school in person. We made the decision to have our son, age 10, vaccinated with the full dose, 30 microgram times 2 BioNTech vaccine in May. I know, shame, shame, shame. Indeed, he had nasty flu-like symptoms, but we have no regrets. Obviously, our son does not have correct official records of vaccination since we provided an incorrect age. He's now eligible for pediatric vaccine, and we wonder whether at some point we will need to proceed with this to at least have official record of his vaccination. I've spoken briefly with our pediatrician, who has a significant number of other parents in the same position. What do you think is the best way to proceed? What would you advise parents in this position? I contemplate possibly having him proceed with 2 official doses, but spreading them out over several months.
Okay, there is an answer to this, and it's a good answer. So if you go to that trusted clinician with whom you have a relationship and you admit that you lied, there is an ability to enter that documentation that then gets uploaded to the state. So here in New York, an individual comes in, they tell me, let's say, for instance, they say, hey, I'm moving back to New York. I've been doing school down in Alabama. We can enter those vaccines into the system. There needs to be a decision by the clinician. Do they trust you? It sounds like you're telling the truth here. And a lot of parents did what you did. You enter that in, and then you enter in when the proper second dose was done, and then you move forward. You don't have to just start giving lots of vaccines to your child to check some boxes. So if your clinician's not aware how to do this, then shoot us an email. But most electronic medical record systems allow you to go in, put in the information, upload it. And then if you come to New York and you use your Excelsior Pass, you're putting in where the information was uploaded from, not necessarily where the vaccine was given.
This is happening because of the ridiculous position the governor of Texas is putting parents in, right? It's crazy. All right, our last one is from Kristen. I have a booster question for you. I'm a healthy 48-year-old U.S. citizen with mild asthma, work in South America. I'm fully vaccinated with Sinovac. Back in April, May, my husband and I traveled to the U.S. in August, and I guess you could say we jumped the gun on the booster advice and went ahead and got a Pfizer jab. No side effects. While we were in the U.S., we wanted to go to events, and our Sinovac vaccines were not recognized. So we got a second jab of Pfizer to be considered fully vaccinated. Now we are 4 jabs in and feeling healthy. Fortunately, the country we live in recognized our U.S. vaccine. Here's my question. Over the next 6 months, I will have to travel internationally at least 3 to 4 times. I also work in an elementary school. This January will be the 6-month mark since my second Pfizer shot and 4th jab. Is there any reason I need a 5th jab? If I do have to get another to keep my vaccinated status in my current country, does it matter what kind I get? Where I live, you typically don't have a choice like you do in the US. You take what you get. I want to be responsible, but is a 5th jab over the top?
All right, so this is excellent, and it really gets at a couple things. So one is, what do we think that third vaccine dose does? Right, because there's sort of this issue here, and one of the things that we think it does is that for a period of time it raises those antibody levels so that they're circulating at a higher level. You're getting a temporary, let's say 3 to 4 month, that's what we're anticipating, vaccine efficacy against infection. Right. So a lot of the third dose, that's been the focus. Recently, there's this suggestion that you may also get a broadening of protection. We don't know if there's protection against moderate to severe disease. So that's one side of this. It's been 6 months. Your antibodies may be starting to drop. Where's that assay? Right. This keeps coming up. It would be great to be able to see that, because there might be a medical reason to go ahead and get that third jab, because it's been 6 months to get that vaccine efficacy against infection for another 3 or 4 months. The other side, is it safe? This scenario you're describing is not uncommon. A lot of individuals have gotten vaccines in other countries or in the same situation you're in. These repeated jabs do not appear to be a problem. There doesn't appear to be a safety issue. So I'll throw all that at you.
That's COVID-19 clinical update number 92 with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you. And everyone, be safe.